Buenos dias, everyone. This is the 3D model of a soul membrane. Let's start. Materials used: scratch papers, shoebox and cutter, bond papers to cover this and this, broomsticks, poster paint, clay and spring, clay, and used folder. Now let's proceed. I traced the edges of the cup or something of the shoebox. Then cut those edges. After that, I trimmed those excess or mess in every side. Now it's done. I plugged in the glue gun so that it will be ready to use in my next step. With those bond papers, I will cover both box and the cup. I used glue gun to stick those bond papers to both box and cup. Compared to ordinary glue, glue gun sticks stronger. Here, I'm covering the cup of the shoebox. The outcome will serve as the support of the other parts. Now it's good. Let's proceed. Using the scratch papers, I crumpled it to form a circle-like shape and serve this as the hydrophilic heads of the phospholipid bilayer. Now it's done. Next, I painted the broomsticks with black and cut them into smaller sizes, and that will serve as the tail for the hydrophobic tails. Then I I stick them in the ends and form V. It may open downward or upwards, and that will serve as our tail. After that, I painted the crumpled papers with green and red color paint. Now that it's done, I now attach those red heads on the box. And those tails. I attached them in both up and down part. Now it's done! Now let's proceed to the green heads. I attach the tail to the green heads and stick them to the outside or outline of our support. Now I'm ready to attach those green heads with tails to the outline. Now we are done with the phospholipid bilayer. With the clay and the spring, I used clay to cover it on the spring, and I used spring to emphasize the helix structure of the alpha helix protein. Now that it's done, it's time to attach it. With the other clay, I made tinier circles and small curves. Yellow clay will serve as the cholesterol. Brown clay tinier circle as the glycolipids. Glycoprotein as small curves with pink clay. And pink tinier circles are the carbohydrates. Now that it's all ready, we are now going to attach it to its corresponding area. I cut the used folder into 3 equal parts. I used one as the protein channel and the other one as the integral protein. Then I painted them both with blue to make it more visible. Now that it's all done, I use the third part of the folder for the peripheral protein. I used tiny sticks to serve as a stand in between the two cutted folder, then used excess paper to cover the sides and cut them away. And I also painted it with color blue. After all, I attached the two other protein to its corresponding area. Now we're done! Let's have a quick montage. The cell membrane, also known as the plasma membrane, is a double layer of lipids and proteins that surrounds a cell. It separates the cytoplasm from external environment. It is a feature of all cells, both prokaryotic and eukaryotic. Cell membranes protect and organize cells. All cells have an outer plasma membrane that regulates not only what enters the cell, but also how much of any substance come in. Thank you for watching.